Well, everybody, thanks for coming today. The impetus for this was in part our admiration for thoroughbreds. They're amazing individuals. You watch them on the track, they're really wonderful. And so it's a bit disconcerting when we read periodically where people say they're on the thoroughbred breed is on the verge of genetic collapse. These are genetic cripples because we're seeing reduced number of starts, we're having catastrophic breakdowns, and this is possible. And I don't know how widely this is accepted, but as geneticists, we find it insulting that our favorite horse is being attacked this way. So we have the culprit in all this is often identified as inbreeding, and inbreeding is responsible for the loss. And so in inbreeding in general, they go through and they just say inbreeding is bad. We should decrease inbreeding. So I'd like to put us on the same page with inbreeding. And an analogy that I like is that inbreeding is like fire. So we use fire to cook and to heat our homes. The other thing is that fire can go and it can burn down our house. So how is inbreeding like cooking? Well, the traits that we value are the products of a large number of genes, each contributing a little bit. And so the process of domestication and selection is a matter of selecting and increasing these genes. There's a loss of diversity as we increase the number of genes that are good and get rid of the genes that are detracting. And that's the process of genetic improvement. Now, how is it like burning the house down? Well, as you can imagine, if we get a deleterious recessive gene in the population, it becomes common, it can undo all the good that we've done. So we're very nervous about these deleterious recessive genes. And so the advice that breeders have often followed is, let's just decrease inbreeding, let's avoid it as much as possible. And it's treated very much as a black box. We don't know where the problems are, so let's not even open the box. So with genomics, we actually have the ability to shed light on the black box. And that was the thing that led us to devise this project a couple of years ago, to go through and just say, can we characterize the diversity that exists? Can we devise a way to manage it and identify sources of problems? So my two colleagues are going to present some of the data that we've been working on. One of them is Ted Kalbfleisch, whom I think most of you know. Ted graduated from the University of Louisville. He went on and got a PhD at Boston University, worked in industry for a few years. He then went to the University of Louisville as a faculty member, and we recruited him away in 2019, and he's been with us since. And he is really the leading bioinformaticist for horses in the world, as far as I think most of us are concerned. He was author of a paper, along with a number of other people in this room, on the most recent reference genome of the horse, and lots of other projects. He's number one in this area, so we're fortunate to have him on this project. The other person in the project is Jessica Petersen, and she's going to also present some of the data. Jessica is, in my mind, the number one population geneticist working on horses. And of special note, a few years ago, she published a paper about the genetic diversity in all horse breeds that I think is a real standard. It continues to be cited in every other paper on horse genetics that I see today. And she's done, of course, a lot of other things, besides that. So we have a really great team to look at the bioinformatics and the population genetics of the horse. So there's going to be two sections. Ted's going to talk about the evaluation of the data, elaboration of the data, and some of the initial observations. Jess is going to talk about some of the population evaluations of the data. Okay.
Thank you, Bernie. Here it is. Okay, so there we go. Okay, so some of you might remember this flyer that went around about 3 years ago. So there was a great deal of concern, at least expressed in the scientific literature, that there was, in fact, too much inbreeding in thoroughbred horses. So those of us who know something about thoroughbred genetics and population genetics, we're thinking about the paper that had come out, and we're pondering, well, how can we respond to this? How can we look into the information they've presented and draw our own conclusions? And there at the time there was no way to do that. Why was there no way to do that? There was no way to do that because there was no publicly available data set that cataloged or quantified genetic diversity in North American thoroughbreds. Well, what to do about this? The answer was relatively obvious, right? You go out, you find as many horses as you can, and you sequence them. You sequence a small subset first in order to catalog the genetic diversity, right? Find out how many polymorphisms are out there. And then you sequence a larger you analyze a larger population to be able to see just what the frequency of these variants are in a large but random sampling of the group that was out there. So our goal was, as I suggested, to generate a public data resource that would do both of those things, catalog and quantify genetic diversity. So the group that we started with was group 2. So this was the population of horses that more or less were born recently, right? So since 2000. Then Ernie, having been a scientist for a very long time, has freezers full of samples that were collected decades ago. Horses that were born between 1965 and 1986. And when he remembered this, he said, well, we've got enough of those that we could sequence those, we could look at those, we could see just what genetic diversity looks like in those animals and compare them to what this population that was born 3 and 4 decades later looks like. So that was essentially the way we ultimately defined the project. We would sequence these 2 populations, and we would use them to catalog and do an initial assessment of individual genetic diversity in North American thoroughbreds. There were 3 phases in this project, and the first was a heroic effort by Dr. Bailey, using guile and charm, and talking to horse breeders and many people who are in this room to get them to participate in this study, to allow us to sample their horses, to do a genetic analysis on them, to answer these important questions. So we sort of set the number. We'd 1,000. There are about 20,000 foals born a year, registered a year. If we could get maybe 1,000 over the last 2 or so decades, that would be a halfway decent sample of what's out there. Ernie was able to go out and collect 1,019. Once we had those, we took a subset of them. So the process that we're using to do this analysis is whole genome shotgun sequencing. Costs about $600 an animal to do this. And we didn't have $600,000. We had about $60,000. So we took a subset of those animals, and in phase 1 to sequence them, and I again established the scope of genetic diversity in thoroughbreds. What's going to happen next is phase 2. Phase 2 is where we are going to analyze the rest of these animals that were collected in order to assess essentially what the frequency of all these variants are, and that is going to give us a marker, right? This is what genetic diversity in thoroughbreds looked like in 2020. Right. So when we think about asking this question again in 2030, we will have a benchmark to go back to and compare it to 2040, 2050, etc. Okay. So how did we select the samples from our group 2? That is the group that was born between 2000 and 2020. from among the 1,019. How did we pick the subset that we got? Okay, so we went and we assembled the pedigrees for every animal in the set that we had, and we used that pedigree information in order to assess the relatedness of the group. Right, the purpose of sequencing was to again define the scope of genetic variability in horses. So find as pick as many animals that were as distantly related to one another as possible to give ourselves the best chance to do that. So Jessica did that analysis. This is what the relatedness plot looks like. All of the blue dots are the samples. The orange ones are the ones that that we selected, and we essentially did that just to pick something from every quadrant as best we could until we got up to over 100. Now we used pedigrees on this, and something that you're because at the time that was all that we had. But what you're going to hear us talk about later is how pedigrees aren't as good as we would like to think that they are for assessing genetic distance between the animals. So that's why I throw the spoiler alert in. We had to do this. If we were doing it again, we'd use the genetic data. Okay, so additional samples for comparison. We had the group 1, which were the 82 animals that Ernie had in his freezer. We had the group 2, that were the 103 horses that we selected from our larger group born between 2000 and 2020. And a researcher by the name of Teruaki Tozaki from Japan published a paper, I think it was in 2021, where he published the genetic variants and the allele frequencies that were found in a population of 101 horses. All we had were, these are the polymorphisms and these are their frequencies. We did not have individual level data. He's recently made that available to us. We haven't had the time to analyze it yet. But these were the 3 groups that we spent most of our time looking at. Okay, what did we do this time that was different from what most folks are going to do? The genetic analyses that, excuse me, many of the genetic analyses that you will have seen in the literature on large populations are done using what's called a SNP chip. It's a wonderful technology that is able to probe in the horse. There are 2 commercial SNP chips available. One has 70,000 polymorphisms on it. The other one has 670,000 polymorphisms on them. And these are polymorphisms that are known throughout horses, not just thoroughbreds. What researchers will do is they will look for variation at those known variant sites. So what is ignored are variants that are going to be specific to thoroughbreds, variants that are going to be low frequency, and variants that are otherwise maybe not as informative as as the people who designed the chip would like them to be. So for example, here, what you see a polymorphism here in this gene MC1R. So this is a polymorphism that would be on the chip. And there are, did it make it here? Any other polymorphism that would show up in the area is not going to be on that chip. So by doing whole genome sequencing, we're able to assess even the new polymorphisms that are showing up in the population, and we're able to quantify those and follow them for decades to come. Okay, so initial results. So from this study, what we've got is that we had 185 thoroughbreds total over both groups. We found 140, or rather 14 million variants in the population. Right. So if you just do a list of single nucleotide variants that occurred in thoroughbreds in our population, it was 14 million. The range in each animal had between 4.4 and 4.9 million single nucleotide variants, with an average per horse of about 4.7 million. And Terowaki, he identified fewer polymorphisms, but the range in individual animal variants was a little bit higher. And in the a paper by Jagannathan and others in 2018, where they looked at 88 horses from diverse breeds, so not just thoroughbreds, they identified about twice as many polymorphisms with a range that's our thoroughbreds fall within. Okay, so variants per horse. What is a heterozygote and what's a homozygote, right? So we've got a maternal chromosome and we've got a paternal chromosome. And when we're looking at polymorphisms, typically what we're looking at are alleles that vary from the reference genome. All right, so the reference genome was built from a thoroughbred mare named Twilight, who's up at Cornell. And so it's most likely that any animal is going to have just, on one of their either their maternal or the paternal chromosome, a variant relative to the Twilight reference. And it's less likely, but it happens that both the maternal and the paternal chromosome are going to vary in the same way from Twilight. And what you see here is, what you see here is that the heterozygous polymorphisms, they double or triple the number of homozygous variants from the Twilight reference. So this is exactly what we would expect to see. And we see that all of the animals are really pretty close to one another with respect to the total number of single nucleotide variants that they have. Okay, so if we look at non-reference allele frequencies across groups, it's important to immediately point out this is an artifact. It's a well-known artifact. So nobody loves the ECCAB3 reference genome more than I do, but it's got its problems. And some of those problems are that regions that are duplicated in the genome, the technologies that we were using to assemble genomes back in the day really couldn't differ. When it saw two copies of something, it couldn't understand whether or not it was represented twice on a single chromosome, or if it was simply represented on the maternal and the paternal chromosome. And oftentimes these duplication events would get collapsed into a single region on the genome. But when the genomic content for those two regions is mapped to the reference, all of that would get collapsed. So what you have is there's some polymorphisms where every animal in the data set is heterozygous, and that's this peak. So that's a well understood artifact. But for the rest of this, what you see is that, so group 1 is are the horses from 65 to 86. Group 2 are the horses from 2000 to 2020, and the gray are the Japanese thoroughbreds. And what we see is that they pretty much lay right on top of one another. The allele frequencies have not changed that much over the years in total. But down here at the bottom, or near zero at the lower frequencies, what you see is that, and this is this region blown up, is that the current Japanese and our group 2, which are the current population, are shifted a bit left to lower frequencies, which is what you would expect. So inbreeding is going to cause a loss of allele frequency in lower frequency polymorphisms. Okay, so one of the Ernie's gonna sum everything up here at the end, but one of the things that he's going to endeavor to impress upon you, and I'll just give you the first salvo. Inbreeding is not the way to assess the genetic health of this population. What we need to be looking at are the accumulation of bad alleles. and we need to be monitoring those, and we need to be mitigating the propagation of those. And what you see in this list are polymorphisms that are known to or polymorphisms that are associated with these phenotypes that these top three are disease, and you can see that they're all at relatively low frequencies, right? 6%, 0.6% in our U.S. thoroughbred populations, and even in the Japanese populations. So they're out there. We know how to look for these things, and these are the things that we should be looking at. There are other genetic diseases that are known that are not found in the thoroughbred, and by that I mean we don't find genetic evidence of these in North American thoroughbreds. This is what's called a principal component analysis, and the way that what we wanted to do here was essentially assess genetically how different were these three groups of horses. So the Japanese horses, the group 2 horses, the group 1 horses. And the principal component analysis essentially it ingests all the variant information that we had for all of these horses, and it endeavors to assess what SNPs allow us to assess the variability between these animals. And the top two sets of polymorphisms are the two principal component analyses that we are principal components that we use. And what those two principal components show us is that the current group 2, the current horses, and the Japanese horse, the green are the group 2 and the red are the Japanese horses, they lay pretty close to one another. And the group 1 horses, the older cohort, is kind of separated out over here and distinct. So the current North American horses and the Japanese horses are more or less moved genetically in the same direction over the years. And at this point, I will turn things over to.
Does that, do you have to correct for population structure in that, or could it be the older horses that were prominent stars at that time, Northern Dancer, and everything was right up front or bold ruler, whereas now they're more distant?
So we use this to look for population structure rather than correct for it, but you're absolutely right in that if there's very high impact individuals in this, they will pull variation to the side. And so I do think that having group 1 over here is a consequence of those horses being fairly instrumental in the diversity that we're seeing now. And so their variation is showing, hey, we're really important, so we're gonna be over here. Because we explain a lot of variation in that data set, if that makes sense. Yeah, Ernie.
Yeah, the group 1, the blue horses, those are horses really from Kentucky. And I wouldn't say they were particularly from a particular line or whatever, but they were very local for this place. They were not a selection group. And this isn't really a good comparison of diversity between these three groups, and we don't want to represent it that way. But that group happened to be very local. The ones that are in green are the group 2 that we selected initially in connection with this study, and they were selected specifically to be as distantly related as possible. So we want that to be done. We don't know how the Japanese horses were selected, and they were over, I think the earliest ones were in the 90s or so.
85 to 2016.
I don't know, but in terms of that 65 to 86 group, that represents a period of time, certainly from the 70s through to the mid 80s there, of enormous transcontinental shift in movement of progress.
Yeah, I mean, and there's population changes. There's all sorts of things. In the 1980s, they were registering 40,000 a year. Now they're registering 20,000 a year. What happened to all those brood mares between now and then? I mean, this is the stuff. Yeah, it's a fascinating thing to do. We don't want to overinterpret the data. But here's the representation. For me, each of us took a different thing away from this. For me, the thing that was impressive was there was substantial overlap in this chart. You know, they were not distinct groups.
Yes,
Jessica was less impressive.
And our data was not yet, phase 2 will be, this is not to reflect the total diversity of the thoroughbred population. What I think is good about this is we set out to sample a great wealth of diversity in those group 2 horses spread across that plot. So we did. And I think Japanese horses overlapping largely, there's similar birth years. So I think
Yeah.
that's what that's reflecting. But I'm going to go on. We come back to that, maybe.
So comparing.
No, I'm going to go on because I'm afraid we're going to really run out of time.
Okay.
But we'll come back to that question afterwards for sure. Is that okay?
Yeah.
Okay. Okay, because I want to talk about this, which I think is more exciting, to be honest. So we're going to talk about diversity, which is what Ernie started with. I love his fire analogy. Because inbreeding is good and bad. We only have thoroughbreds because inbreeding was performed when the breed was founded to select horses that were really specified for what we wanted them to do. And same with any other breed that we have. Inbreeding is how we get breeds, right? So now we're just thinking about, well, do we need to worry? So I'm going to talk a little bit about how we measure inbreeding using genomics. Going to make an argument that pedigrees are super tools. They help us find horses or look at relationships, current relationships among horses, but they don't, they're not giving us the and then a little bit about how these data can be used, and then Ted's going to expand upon that at the end. Okay, so I'm just going to walk through this because this is how we think about inbreeding using genomics. So on the left, pretend that's a sire, that's your favorite stallion. He has 31 chromosomes, an X and a Y. So I'm just representing one pair of chromosomes. He got one from his dad, the paternal chromosome, one from his mom, the maternal chromosome. I'm using colors to represent where they originated so you can track them. So he was bred to a mare. He gave his genetic material to that foal. So that foal is half of his genetic material. This is blue and yellow because there's recombination that happens, and he gives part of his dad's genome, part of his mom's genome to that foal. That mare gave the other half of that foal's DNA to it. So that chromosome is black to show that it's not related. When we bred the stallion to another mare, he gave his mom and dad's genome to that foal, and the other mare gave her genome to that foal. But notice that that stallion never gives exactly the same parts of its genome to that foal. But now we have half siblings, right? And we know that inbreeding is caused by mating between relatives. So if I mate these two horses, I'm going to mate between siblings, and I expect to have inbreed. What inbreeding is when this horse and this horse each give genetic material to their new foal, this baby, there's going to be parts of the genome where each chromosome is identical, right? So we think about a recessive, that's when recessives happen. When there's two alleles that came from the same source that come together. So this is what we call a run of homozygosity. So homozygosity just means they're the same genetics. And that length of that run of homozygosity is going to be pretty long because these guys are pretty highly related. So they're going to have a lot of their genome that is going to be homozygous that they're going to share that originated with from this initial stallion. Good. Okay, so what if instead we didn't breed these two half-sibs, but we bred several generations from that foal, we bred several more generations here to unrelated individuals. We end up with two foals here that are going to have a variety of genetics from whomever was bred in the meantime, but they are both great grandchildren of our initial stallion. So they're still related. They share genetics, and if we breed these guys, we're going to have inbreeding. But this inbreeding is coming from 6 generations back in this case. When that happens, we still get runs of homozygosity because mom and dad were related, and they can give that foal the same chromosome, the same genetics. But those runs of homozygosity are shorter, and there's fewer of them. So I'm going through this because the length of the run of homozygosity gives us a hint as to how recent that inbreeding was. If it's really long, inbreeding was recent. And if that run of homozygosity got broken up just due to recombination and all these different genetics being infiltrated, that run of homozygosity is going to be very short. Okay, so I said I'm going to tell you why genomics can give us more information than pedigrees. So we're looking into that black box that Ernie mentioned. So challenges with pedigrees. Challenge number one is when we use pedigrees to estimate relationships between two individuals, we have an assumption. One of the assumptions is that an individual inherits 25% of his genome from each of his grandparents. We know that's not the case because it's random what part of the genome that parent, each of those parents, is going to give to that full. Another example, we expect that full siblings are related at 50%. I spit in the tube for 23andMe. My brother and I are only related at 46.5%. Okay. So if we can't estimate relationships well, we can't estimate inbreeding in the progeny of those individuals very well. Oh, sorry. So if we can estimate relationships well, that still doesn't tell us what parts of the genome two animals share. And it doesn't tell us what parts of the genome are a result of inbreeding, right? We could know that this animal is supposed to have 12.5% inbreeding. We don't know if it's on chromosome 1, on chromosome 2. So genomics can help us answer that question. And finally, the other challenge with pedigree is it has this assumption that all of our founders were unrelated and themselves had no inbreeding. We know that's just not true because there was not a population of horses that started the thoroughbred breed that were totally unrelated to one another. So our little expectation for pedigrees is that this sire has two chromosomes that were totally different because the founders were unrelated, but we know that that's not the case. There's probably inbreeding there, and those founders were related. So those are challenges with pedigrees that we can do better using genomics. So now that I said that, I'm going to show you our comparison of estimates of relationship using our genomic data compared to what the pedigrees say they should be. Okay. So on the x-axis here is a real estimate of relationships using our sequence data. And on the y-axis is that using our pedigree data. Okay, the line is what we'd expect if they are perfectly correlated. If our pedigrees were telling us exactly how much genomic information was shared by two individuals. So up here we have actually a mother-daughter pair. And so it has to be that mother gave her daughter 50% of her genome. We're pretty dang close to that 50% line, and our pedigrees tell us that as well. But as we move down, here, we're not doing a very good job of estimating relationships using pedigrees. So these horses, this horse here shares about 20% of its genome with another horse. The pedigree says it should be unrelated. Okay. So we can use genomics to do a better job. So if we don't do relationships well, we don't do inbreeding well. So this is the similar figure, except I flipped axes, I apologize. So if pedigree-based inbreeding coefficients here, and this is our genomic based inbreeding coefficient here. This is our line that we would expect if they were identical. So you'll notice that our inbreeding estimates based upon our genomics are much, much higher than what our pedigrees say. And they're also just not very well correlated. They're not really sitting on a line. So I could have a horse here that the pedigree says should have 2% inbreeding, and it could have anywhere from 26 to 35% inbreeding or homozygosity in its genome. So we can look at our inbreeding based upon how big those runs of homozygosity are. So that's going to give us a clue as to how recently that inbreeding occurred. So on the bottom are runs of homozygosity that are relatively small, less than 1 megabase in size. Each of these vertical lines is an individual. So this first one has about 15% of its inbreeding coefficient that came from short runs of homozygosity. Another about 15% that came from kind of medium runs of homozygosity, 1 to 5 megabases. And then we really see that we're not seeing many at all of those long runs of homozygosity that we would expect if there was very recent inbreeding. And really, if you look at thoroughbred pedigrees, there's not very recent inbreeding. People aren't breeding captives to one another, and they're not breeding sires to daughters. So these are separated by the groups. This is group 1, this is group 2, by year of birth. So we see that there's some little yellow peaks that show some long inbreeding, but I would argue that they're really not any more prevalent in group 2 than they are in group 1. And like Ted showed, with his variance figure, they're pretty similar across the whole data set. Yeah, Bernie.
Jessica, can you comment on why the length? You know, the timing is important. I mean, the idea of bridging selection, the shorter fragments have been around longer, so.
Yeah, so
Quick question.
there's a question in the audience of what does it mean about the length of the fragments and how long they've been around. So when I started out by saying that inbreeding was a reason we have thoroughbreds, there's probably a lot of parts of the genome that are homozygous where there's inbreeding, because that's what makes a thoroughbred, right? And as generations go on, if there is something deleterious in those, they can get selected out. Breeders have done a good job of selecting out bad stuff. And so we're going to eliminate those big runs of homozygosity that have some of those negative things in it. And these shorter runs of homozygosity are less likely to be detrimental. They're likely to have been purged of deleterious information. So good, Ernie? Yeah. So if I average this by groups, if we just want to compare our two different groups, we can see that again, that really most of the inbreeding we're seeing is short runs of homozygosity. To me, that means it's old, that it's inbreeding that happened many generations ago. And there's really not a lot of recent inbreeding that's showing up. And it's really not any different between their two different groups that are separated by 4 generations. And where they are different is in these shorter runs of homozygosity. We can look at it by year of birth. So this is just the data we have. We haven't densely sampled enough to draw concrete conclusions about this. But you can see that so the individual dots are individual horses. The yellow diamonds are the average per year of birth. They're kind of bouncing all over, and there's a lot of overlap in what we're seeing in our groups. If we're looking at the rate of change of inbreeding, it's not different between those two different groups based upon these data. And again, especially in this group 1, some of these years of birth are only represented by 1 animal. So it's not a good data set to draw robust conclusions about that. But when we think about how we can use these information to monitor our horses, I think this is a really cool example. So this is looking at the proportion of markers and runs of homozygosity across chromosome 29. I don't know why I chose that one, but I did. So this is group 1, and you can see that kind of in the middle of the chromosome here, there's a lot of horses that have a run of homozygosity. And over here, there's another section where there's a lot of homozygosity. Those might be targets of selection. There might be important genes there that are contributing to athletic performance. And it's kind of nice. We see similar peaks over here in group 2. And I can overlay that. So this is group 1, it's going to be in black. And group 2 now is in red. And we can use this to identify regions of the genome that have been changing over time due to selection, due to drift, all those other forces that you'd have to investigate. So black was group 1. So whatever this region is here has increased in frequency in group 2. And then over here, this region that was homozygous in quite a few horses in group 1 is now less homozygous in group 2. So this is information that we can use as we track horses and we track the breed going forward to look at how breeding practices are changing the genome. And so Ted's going to take over now, and he's going to tell us more about how we're going to use these data for phase 2 of the study.
Yeah. Thank you. Okay, so now that we have genetic data on a lot of horses, what we can do is we can build what's called haplotype structures, right? So in the pictures that Jessica was showing a little bit earlier, you saw that from generation to generation, you had big chunks. single bases do not move from parent to child. Big chunks of DNA are what are being passed a parent to child, right? And those are referred to as haplotypes. And if we can build ourselves a catalog of these polymorphisms that are moving together, right? What we can do is we can create a database that will tell us, if I have this allele, I know that in this megabase of sequence, I've got all of these other alleles as well. So this is something that's being done well in cattle and in human genetics. It's referred to as low-pass sequencing, and essentially what it allows you to do is what we sequenced in this population was about 20-fold. That means there were about 20 reads per every base in the genome. With low-pass sequencing, you can sequence about 1-fold coverage and still impute whole genome genotypes. Why would you do this? You go from $600 an animal to about $50 an animal to generate whole genome genotypes. There's a lot of stuff that you're going to miss, but it's going to give us the majority of the information that we're going to need to accurately track the change in gene frequency over time. What we've shown is, this is the information that we have for decades ago. This is the information that we have on a few contemporary animals. We'll be able to flesh this out with another 900 animals, and we'll be able to continue to generate data well into the future to work out exactly where the breed is going. And modeling. So can we guess at changes in genetic diversity as a function of time? All right. So if you were to take a stallion and you were to mate it to 100 mares, right? You could more or less predict just what the frequency of the alleles are that animal is going to pass on just by simple math, right? So if a stallion is homozygous for one allele and the mare is homozygous for one allele, you know that the foal is going to be homozygous. If the sire is homozygous for one allele, the mare is homozygous for another allele, you know that the foal is going to be heterozygous. If the mare is heterozygous and the stallion is homozygous, you know that 50% of the foals, or 50% of the foals that those two animals would produce would be heterozygous, and the other half would be homozygous. And then the same for compound hets. You can do the math and figure out just what's going to be passed on. You can do this for the whole genome, right? And you can do this for every mating. And as a result, what you could easily imagine doing, right, is suggesting to your simulation software different breeding strategies, right? So if I'm covering, if a stallion is covering 10 mares, what does genetic diversity look like going forward? If a stallion covers 100 mares, what does genetic diversity look like going forward? If you're simply doing random matings, what does genetic diversity look like going forward? And you can predict trends that you can subsequently compare actual data to see just in what direction the breed is going. Okay, and your plots look something lovely like this, where you would see oscillations, and again, you would look to see what the real measures are, and you would compare them to these predictions. So what is it that we're doing next? Well, I think the most substantial thing that's coming out of this project is the data that we have used is being made publicly available. That's happening on August 31st. We have created a benchmark, and we can now continue to generate new data for comparison and breed level evaluations. Building a haplotype map to aid in low-pass sequencing, that is something that is going to help the thoroughbred breed immensely by allowing us to generate whole genome genotypes really very inexpensively. So Ernie, I'll turn it over to you now.
we're quite keen for your responses actually to this. I know that there's a lot of data that's here. We've been looking at it for months. We've been talking about the presentation for weeks, and we've dumped it all on you, like that. And everything may not resonate with you. We're interested in what questions you have. On the handout that was here, I hope people got the handout. Got a list of summary points that I believe that we have made. And that's the question we have, is how well do you agree that we have made the points here? And then there are some recommendations. There's 5 recommendations that we have going forward. And so I'm just going to walk through, and what we'd like to do is, if you have a question ready at the end of this to say, I don't believe that, or how did you get there, or what was the evidence for this? So one of the things I think as we were saying earlier, pedigrees are great. We don't want to take away from that, but pedigrees are useful to identify the individuals that you want in your genome, but they don't measure the homozygosity, or I mean, it's a black box. We don't know what it is, and genomics can be used to do that. So we're advocating using genomic measures if you really want to know how much homozygosity there is in there. The number of genomic variants, Ted showed the comparison with the Japan data and with the group from Switzerland that had the 88 different breeds of horses. And the thoroughbred is low in diversity, but still falls within it. So it's not a clonal animal. There's still quite a bit of diversity there. Again, the caveat being that these are selected populations. So we're waiting to go through to phase 2. Cluster analysis, this is looking at the overlap, the PCA analysis, and the relationship of the Japanese and the group 1 and the group 2. And they were not far apart from each other. There was considerable overlap. We had quite a bit of discussion about how to talk about that, but they weren't really that diverse. The population, the thoroughbred is a worldwide population, so this wasn't surprising. These are probably the main points here. Most of the inbreeding that we observed in the thoroughbred, and this was really news to us, was with these short regions of homozygosity. And as Jessica said, the short regions of homozygosity, they occurred generations ago. They're small, which is why that's why they're small. But the other consequence of them being generations ago is that breeders have had time to select for them. This is what the traditional genetic advice is. Let's practice inbreeding slowly so we can select and purge the deleterious genes. Most of the regions of homozygosity that we found, I mean, the lion's share of them were short or medium. Breeders have done a good job, and I bring up, there was a fellow, Ludovic Orlando, who did a study and was comparing genetic load, deleterious genes in different breeds of horses, and he was surprised to report or discover that it looked like, from the way he looked at the data, that the thoroughbred had a low level. It looks like breeders have been doing a good job, and that would be consistent with other. The ones that we want to be concerned about are the long runs of homozygosity. They represent things that have recently been introduced, and those are the ones that, when you look at other breeds, potentially harbor deleterious genes. And so one of the things that we're advocating is, in a surveillance program, let's identify these, let's investigate these before they become too common. And frankly, if they're already regions of homozygosity, they become fairly common. These are the ones that we should focus on. And then the last one I'll do here is we can use genomics to precisely identify things. Ted showed that one chart is that there's a number of genetic diseases that have been identified, and we can test for them. If we have the whole genome sequencing, we can do that. We can't do that with the SNP chip data that has been used. We are making all the data available. That was one of the things that was frustrating at the beginning, was that we said some of the things that people were reporting didn't make sense. We had no way to interrogate it. We had no way to go back and do it without generating our own data. So we generated our own data. We also want to make it available so all the other smart people around the world can look at the data and also make discoveries. And again, as Ted says, we're going on to the next phase. We've got 900 other animals in the bank. We may go out and get some more. We want to sequence them and then do a proper population study and ask things like, what are the consequences of the changes in numbers of horses over the last 4 years? What are the changes in potential breeding practices? So there's a lot of interesting things that we can do with the modeling. And then there's recommendations. I think we've already said those. I would be more interested in opening it up to questions, and this is on the sheet. So, conflict of interest. Ted wants to make sure that you know he has a conflict of interest. He's a very honest fellow. He has a company that does something, but that may be related to this, but there we are. Ted isn't happy with my elaboration, but anyway, we have acknowledgements. The Kohler Foundation from the Kohler Fund from the Gluck Foundation supported the work that we've done up to this point. And we're very grateful to the breeders who generously provided the samples. And I don't think it was said, the samples are anonymous. I mean, we got information about them. We use them for pedigrees and all that, but we've got this. It's being kept very confidential. No one is going to know who the horses are. They are just a random population of horses.
It doesn't look like a thoroughbred farm.
Pardon?
It doesn't look like a thoroughbred farm.
I know, and I dropped this in.
That's a test of him, but
This is one of my favorite pictures, but I felt bad that it wasn't a thoroughbred farm. But yes, this is in Utah. This is a wild horse herd. So with that, questions that you've got.
we've got a couple of online questions.
Let's start, Naveed.
So, I was thinking like the group 1 versus group 2. One of them was selected to be fairly as diverse as possible, the other one being very local. So when you're comparing the genetic diversity of these two groups, don't you think that would introduce kind of a bias or, kind of?
So,
So, the question needs to be repeated. You want to come up and repeat the question?
sorry. Okay, well, I'll come down here. So the question was, we selected group 2 to be very diverse. Group 1 was opportunistic, what was in the freezer, and could that influence our results? So Naveed, you said the population diversity. We're not saying anything about population diversity at this point. Our diversity analyses were individual levels of inbreeding. And so those individuals don't represent the population accurately. Neither one of them, neither group does, but those individuals represent their own diversity perfectly. So I agree that the sampling method would bias what we're seeing if we're comparing them as populations, but I want to be clear that we're not looking at populations right now.
Our first step really was to look for diversity. And it was an item of curiosity about how diverse, what would the differences be in those the two groups. We're presenting the data not so much to say, here are the population differences, because we can't do that with these small numbers and the differences in the collection, but we were quite struck at actually how similar they were.
Yeah, and the goal,
In a qualitative sense.
the goal from this phase 1 was to set up phase 2. So to be able to impute those haplotype blocks well, we need to know what diversity exists. So that's why we went after those samples that represent, based on pedigree, which is the best we had, as much diversity as possible. That makes sense.
And that diagram that we showed about, the selection of animals based on pedigrees with those orange dots being the animals that were picked. How are they scattered in terms of group 1 versus group 2? Are they does group 1 capture the whole diversity as well as group 2?
That was just group 2.
Oh,
An online question?
Yes, so the online question is from Dr. Reed, and he's asking, how can you use these techniques to determine what role genetics plays in a particular problem? For example, cervical vertebral stenotic myelopathy or equine degenerative myeloencephalopathy. And if you are looking at this particular problem or for a particular problem and interested in keeping costs low, can you use the lower cost technique that you described, the low costs? I'm probably speaking.
Yeah, so that's the hope. What this gives us first off is a large data set with which we can assess allele frequencies, right? So if you see alleles in a particular in a horse that is affected with whatever condition that is thought to be genetic, right? Variants that animal has that occur at 60% allele frequency in the population are likely not going to be the ones that are responsible for that condition.
This is a great place for you to tell them about your other project with the PDL.
So we've already used this data set as a control population. Some of you who may have been here to hear me pitch a project to the Gluck board about sequencing idiopathic abortions. Right, so there are any number of polymorphisms that we found that only occur as heterozygotes in 183 animals, right? Some polymorphisms, no homozygotes, and your gut instinct as a geneticist to say, that's probably a recessive lethal or something like that. Well, almost all of them are not, right? Almost all of them are just victims of bad statistics. So what we did was we sequenced these idiopathic abortions to look for homozygotes in those animals that do not occur in the larger population. And we've found several candidates that are quite promising. And that is using this as a control population. So already the data set is useful, and once we've generated data for another 1,000 animals, we'll have a much more powerful tool. Yes, ma'am.
How concerned would you be about sequencing errors with the low pass sequencing for establishing?
Repeat the question.
A wonderfully intuitive question, right? How concerned am I going to be about sequencing errors in the in low pass sequencing? So I wouldn't be concerned about sequencing errors per se. What I would be concerned about are imputation errors, right? So what you're not going to get an accurate characterization of very low frequency alleles. You're not going to get a good characterization of new alleles. And you have to go into any project understanding that completely. So if you're looking for a disease that has been a genetic disease that's been in horses for years, for centuries, it's going to capture things well. If it's something different, if it's something newer, or if it's some de novo mutation that you see in a new foal that's done something that's never been seen before, that animal you're going to have to sequence to 20x. Yes, sir.
So this is short read data.
Yeah.
How much variation in the, we don't know this in the horse, but say in the Welsh and humans, how much variation has been attributed to copy numbers versus sequence differences?
A lot. I can't quantify it any better than that, but a great deal. I mean, they're just large chunks of the genome that are duplicated or that's happening a bunch, and getting a handle on that is really very hard. Right. So when I was talking about those polymorphisms that show up only as heterozygotes, right? If you've got any duplicated region, especially one that's not fixed in the population, it's only going to show up as heterozygotes, and you're going to think that it's a deleterious recessive, but it's not. It's simply a copy number variant, and that there are thousands of those that you see. Short read data is the best tool that we have right now for the money that we've got. Our sequencing technology and our sequence assembly technologies are improving day by day, and I'm sure that in 5 years we're going to be doing something different.
We have another online question from Dr. Reed, who asks, how can this information be correlated to Jockey Club data on race performance of horses from, say, 1970 to today?
Anyone?
So the question is, how can it be correlated with race performance? Honestly, it can't, because these horses are anonymous and we're not associating phenotypes with them. So I guess I can't answer that any differently.
But so this data, no. But if that was something somebody wanted to do, and if they, could get the samples, it's absolutely possible. Whether there would be any utility in doing it, I can't say.
It's possible to ask question, you
Sure.
don't know whether they'll be associated.
Correct. Yeah. Yeah, but yeah, a lot of things have to happen, like you've got to be able to get the sample, you've got to, yeah. Yes, ma'am.
So to follow up with that, can you explain again what you were showing us with the chart that had the group one horses having a lower proportion of the sprinting tendency?
Well,
That was, let's make sure we're talking about the same.
It's not like that's not a group one of the mistakes. Right.
Is it,
I understand.
It's the
Yeah.
frequency. That one, that one.
this one?
Yeah.
yeah, so
The sprinkle was lower.
Yeah.
yeah, so it goes back to this is a random sample divided population, so I wouldn't really take that as it's significantly changed or not.
Up until that last question, I was taking. Solid.
Yeah.
But I mean, that is, you know. Selection biases aside, it is interesting that frequency has changed in excess of 10%. Yeah, that does get one's attention.
Just based on the results that we got out of phenotyping for the MSD and sign insertion, looks like that the newer generation is going towards the tendency of having more sprinting ability.
Yeah.
This is just the preliminary data that we have from other people.
And you don't know if Ernie's horse is better than her horse,
Right.
you know, so.
Yeah.
Other questions? No, thank you for coming. And if you've got questions or insights, we would be grateful to hear from you because it's a large data set to ponder. It's interesting questions we're pondering. Thank you.